There's a story going around. A secret laboratory in Alaska. 4 capsules containing an ancient, deadly virus have disappeared from the permafrost. Is this true or fiction? We started investigating, and what we discovered was more terrifying than any legend, because the real threat is no longer sleeping under the ice, and it's much closer than you think. In the most remote and coldest corners of our planet, where civilization gives way to permafrost, secrets lie hidden that could change the course of human history. One such story, like an icy wind, swept through the darkest corners of the internet, gathering frightening details and causing alarm among all who encountered it. It begins in Alaska. Imagine a secret research complex lost amid endless snow. This is not just a laboratory. It is a 4th-level biosafety facility, an outpost of science at the edge of the world. Its mission is to study what has been hidden in the ice for millennia. Scientists here work with unique samples extracted from the depths of the permafrost. Mammoth tissue, the remains of ancient animals, and, most importantly, microorganisms that were frozen before the dawn of modern civilization. These are ancient viruses unknown to science, true time capsules from the prehistoric past. On one unremarkable polar day, when a blizzard raged outside and the sun never rose above the horizon, the unthinkable happened. At exactly 2 o'clock in the morning local time, the complex's security system failed. For several minutes, all the surveillance cameras went dark, and the electronic locks on the doors of the most dangerous material storage facility were disabled. When the system rebooted and the security shift finally reached the cryogenic unit, they were met with a shocking sight. The door to the storage facility, protected by several levels of access, was ajar. Inside, there was a cold, dim light. But that wasn't the worst of it. The special box containing 4 capsules with the most promising and dangerous samples of ancient pathogens was empty. 4 capsules. 4 unknown viruses that had been dormant in the ice for tens of thousands of years had disappeared. There were no signs of a break-in, no fingerprints. The cameras had recorded nothing. It was as if the shadows themselves had carried away the deadly contents of the laboratory. This story, passed from mouth to mouth on online forums and anonymous blogs, painted an apocalyptic picture. What were these viruses? Were they capable of infecting modern humans? And who, or what, was behind this daring and silent theft? Government special services, a rival corporation, bioterrorists, or something even more mysterious? The legend of the ice laboratory became a symbol of our deepest fears. Fear of the unknown that lurks in nature, and fear that the technologies created to protect us could turn against us in an instant. But is there even a grain of truth in this chilling story? To find out, we must embark on a journey that will take us from the snow-covered wastelands of Alaska to the scorching shores of Australia. From real scientific discoveries to the depths of human psychology. This is not a story about what was lost, but about what we are afraid to find. When a frightening story gains enough detail, it ceases to be just a rumor. It becomes a challenge. For a small group of independent journalists and enthusiastic researchers, the legend of the missing viruses from Alaska became just such a challenge. The initial excitement and anticipation of a scoop quickly gave way to tedious and seemingly fruitless work. They began by combing through official sources. Dozens of requests were sent to leading U.S. scientific and government agencies with ties to Alaska. These included the Centers for Disease Control and Prevention, The National Science Foundation, the University of Alaska Fairbanks, which does indeed conduct research on permafrost, and even military agencies with facilities in the region. The responses, when they came, were discouragingly uniform. In response to your inquiry, we have no information about any incident involving a biosafety breach or the disappearance of biological materials from laboratories in Alaska. No reports, no official investigations, no mention in the press. A wall of silence. But was it a wall of conspiracy or simply a wall of nothingness? The researchers went further. They began analyzing satellite images of remote areas of Alaska in search of unmarked complexes that could match the description of a secret laboratory. They interviewed former research station employees, small aircraft pilots, hunters, anyone who might have seen something unusual in the Arctic wilderness. The result was zero. No one had heard of such a facility. The story began to fall apart at the seams. All the details that had seemed so convincing turned out, upon closer inspection, to be too cinematic, too perfect. Cameras turned off for exactly a few minutes. A mysterious disappearance without a trace. It all sounded more like the plot of a Hollywood thriller than a real event. Disappointment grew. Was it just a hoax? A fake created by someone for fun or to cause panic? But then, why did it resonate so strongly with thousands of people? Why was it so easy to believe? The answer to this question lay not in the realm of facts, but in the realm of psychology. The story worked because it exploited real, legitimate fears. Science is indeed encroaching on a world that has been frozen for millennia. Permafrost is indeed melting at an alarming rate. And accidents in laboratories, albeit not as dramatic, do happen. It was at this moment, when the investigation had almost reached a dead end and was about to give up, that one of the researchers, looking through news archives for the past few years for the keywords disappearance, virus, and laboratory, stumbled upon something striking. The news was not from Alaska, but from the other side of the world, Australia. Although the location was different, the details of the incident were eerily familiar. There was also a laboratory there. There were also missing samples, and there was also a long, inexplicable silence from the authorities. This was the first real lead. The key to solving the mystery of the ice lab lay not in the snows of Alaska, but under the scorching sun of Queensland. The investigation took a new and unexpected turn, shifting attention from the fictional drama in Alaska to real events in Australia was like stepping out of a dark movie theater into bright sunlight. Vague rumors gave way to documented facts, official statements, and a real, not fictional scandal in the scientific community. The event that became known as the Queensland incident lacked Hollywood glamour, but that didn't make it any less disturbing. On the contrary, its ordinariness and bureaucratic undertones were even more frightening than any conspiracy theory. It all began not with the cameras being turned off, but with something much more mundane, an audit. In August 2023, employees of the Queensland Department of Health conducted a routine inventory at the State Virology Laboratory. This institution was responsible for the diagnosis, research, and surveillance of various pathogens. When the auditors reached the cryogenic storage facilities, they encountered a glaring discrepancy in the records. Hundreds of samples that should have been in freezers at ultra-low temperatures were missing. They were not on the shelves or in the records. They had simply vanished from the system. Alarm grew with each passing hour as the scale of the disappearance became clear. We were not talking about 4 test tubes, but 323 containing live viral cultures. This was not just a breach of protocol, it was a biosafety disaster. Queensland's Minister of Health was forced to make a public statement, calling the incident a serious historical breach of biosafety protocols. But the most shocking revelation was yet to come. During the investigation that followed, it emerged that the incident that led to the loss of the samples did not occur in 2023, but 2 years earlier, in 2021, for 2 years, no one noticed that hundreds of test tubes containing deadly viruses were missing. For 2 years, one of the most secure laboratories in the country had been operating unaware of a hole in its security system. This fact was more astonishing than any story about mysterious kidnappers. It pointed to a systemic failure, to human error, to negligence that could have led to unpredictable consequences in such a sensitive area. A story that had previously been nothing more than a shadow, an echo of fiction about Alaska, took on flesh and blood. The names of the missing viruses became known, and these names sent shivers down the spines of not only experts, but also the general public. The list included the Hendra virus, lyssavirus, and hantavirus. Each of them is dangerous in its own way and capable of causing serious, often fatal, diseases in humans. The parallels with the Alaska legend were obvious. Secret work with dangerous pathogens, their sudden disappearance, and the long silence of the authorities. But there were also key differences. There was no mystery here. There was only the disturbing reality of a bureaucratic error and the potential danger released into the world not through malice, but through simple carelessness. And this reality demanded the most careful investigation. What really happened within the walls of the Queensland laboratory in that fateful year of 2021? To understand how such a massive loss could have occurred in an institution that, by definition, should be a model of order and security, investigators had to reconstruct the events of 2021. The picture that emerged from memos, emails, and employee statements was a story of a cascade of failures, where one small problem led to another, ultimately resulting in chaos. It all started with a trivial technical malfunction. One of the ultra-low temperature freezers, operating at -80 degrees Celsius, broke down. This in itself is not an emergency. Laboratories of this caliber always have backup equipment and clear protocols for such an event. The protocol required the immediate and carefully documented transfer of all biological materials from the malfunctioning chamber to a working one. Laboratory staff began to carry out this task. However, it appears that something went wrong at this point. The samples were moved in a hurry. Perhaps it was the night shift, or perhaps there was a staff shortage. The main mistake was that the transfer was not properly documented. No transfer reports were drawn up, and no relevant entries were made in the electronic logbooks. Hundreds of small test tubes, each containing a potential threat, were transferred from one safe to another without a proper inventory. This was a gross violation, but it did not yet mean loss. The samples were simply not where they were supposed to be according to the documents. But then, the situation got worse. A faulty freezer, which was now listed as empty on paper, had to be disposed of. And here lies the second, possibly fatal mistake. It is possible that some of the samples were never moved. They could have remained in one of the boxes or containers inside the broken freezer. In this case, their fate was sealed. They were disposed of along with the old equipment, most likely destroyed in an autoclave, a special oven for sterilization. This would have been the best of the worst scenarios, as the viruses would have been guaranteed to be destroyed. But there was another, more disturbing version. Some of the samples could have simply been lost in the rush of moving from one chamber to another. The test tubes could have been accidentally thrown away with regular laboratory waste that had not been sterilized. Or, although it seemed highly unlikely, someone could have simply taken them. The investigation report contained a dry but frightening phrase. The materials may have been removed from the secure storage facility and lost, or their location could not be determined for other reasons. This wording left room for a wide range of interpretations. It pointed to a complete failure of the accounting and control system. The laboratory had not simply lost the viruses. It did not know how, when, or where exactly this had happened. It was a black hole in the biosafety system. And in this hole, 323 test tubes containing pathogens capable of causing deadly diseases disappeared for 2 years. Now it was necessary to understand how great a threat was contained in these small, lost vessels. To fully appreciate the seriousness of the Queensland incident, it is necessary to understand what exactly was in the missing test tubes. These were not abstract, hazardous substances. They were specific, well-studied, but no less formidable enemies of humanity. The main hero of this list was the Hendra virus. Of the 323 missing samples, nearly 100 contained it. The Hendra virus is a relatively new pathogen, first discovered in the mid-1990s in Australia, following an outbreak of a mysterious disease among horses in the Brisbane suburb of Hendra. Its natural carriers are flying foxes, large fruit bats. The virus can be transmitted from them to horses and then from horses to humans. Direct transmission from bats to humans is considered extremely rare. The Hendra virus is extremely dangerous to humans. It causes severe respiratory disease and neurological complications, in particular encephalitis. The mortality rate is estimated to be between 50 and 75%. Only a few cases of human infection are known, but most of them ended tragically. A vaccine exists only for horses. There is no specific treatment or vaccine for humans. The loss of nearly 100 samples containing this virus was a nightmare for epidemiologists. Next on the list were 223 test tubes containing viruses. This is a group of viruses that includes the well-known rabies virus. Australian bat virus, as the name suggests, is also transmitted by bats and can cause a disease in humans that is virtually indistinguishable from rabies. Without immediate vaccination after a bite or scratch from an infected animal, the outcome is almost always fatal. Finally, 2 test tubes contained hantavirus. Hantaviruses are transmitted by rodents and can cause 2 serious diseases in humans: hemorrhagic fever with renal syndrome or hantavirus pulmonary syndrome. The latter, characteristic of viruses circulating in North and South America, has a mortality rate of about 38%. Although there have never been any reported cases of hantavirus in humans in Australia, the presence of live cultures in the laboratory posed a potential risk. The very nature of these viruses made the situation particularly alarming. All of them are zoonotic, meaning they can be transmitted from animals to humans. This means that even if the missing samples had entered the environment and did not directly infect humans, they could have infected local animals, rodents or bats, creating a new, uncontrollable natural source of infection. It would have been a real Pandora's box. However, paradoxically, it was the nature of these viruses that offered the main hope. They are all quite fragile and do not survive well outside their host organism or the special conditions of ultra-low temperatures in a laboratory. In the open air, exposed to ultraviolet light and heat, they quickly break down and lose their infectivity. This fragility became the main argument of the authorities trying to reassure the public. But the question remained, was it possible to completely rule out the scenario in which at least one of the 323 samples survived and found a new host? When information about the missing viruses leaked to the press, the Queensland Department of Health was hit with a wave of criticism. The main question asked by journalists and the public was not so much what happened as why are we only finding out about this now? The 2-year delay between the incident and its discovery undermined trust in official structures and gave rise to a wide variety of theories. The authorities found themselves in a difficult situation. They needed to acknowledge the seriousness of the failure, conduct a transparent investigation, and at the same time prevent mass panic. The Chief Medical Officer of Queensland, Dr. John Gerard, became the main spokesperson for the health system. His task was to provide the public with a balanced assessment of the risks. The main thesis of the official position was that the risk to the population was extremely low. This optimism was based on several key arguments. First, as already mentioned, the fragility of the viruses themselves. Gerard repeatedly emphasized that the samples would have broken down very quickly outside the freezer and become non-infectious. It is difficult to imagine a scenario in which the public could be at risk, he said at a press conference. Second, the authorities insisted on the most likely disposal scenario. Most likely, the lost test tubes were destroyed in an autoclave along with discarded equipment, which is standard procedure for all laboratory waste. It's just that this process wasn't documented. Third, epidemiological surveillance didn't find anything unusual. Over the past 5 years, there have been no reported cases of Hendra virus or lyssavirus in humans in Queensland, and there have never been any reported cases of hantavirus in humans in Australia. The absence of outbreaks was indirect, but compelling evidence that the viruses had not entered the environment. In response to the incident, the Queensland government launched a major internal investigation. Its goals were not only to determine the exact circumstances of the disappearance, but also to review all existing policies and procedures in the laboratory. Staff retraining, an audit of all permits for the storage of hazardous materials, and the implementation of stricter accounting systems were carried out. However, despite all these measures and reassuring statements, skepticism remained in the expert community. Dr. Sam Scarpino, director of the Department of Artificial Intelligence and Life Sciences at Northeastern University in Boston, commenting on the situation, confirmed that the missing samples contained deadly viruses. Although he agreed that most of them are not transmissible from person to person, which reduces the risk of an epidemic, he called the very fact of such a loss in a laboratory of this level unprecedented. The Queensland incident was a harsh lesson. It clearly demonstrated that the greatest threat when working with dangerous pathogens is not sophisticated malicious intent, but the ordinary human factor: fatigue, carelessness, and violations of routine procedures. And while officials in Australia were trying to deal with the aftermath of this lesson, our investigation, which began with a fictional story in Alaska, was ready to return to its origins. After all, the second part of the legend, about ancient viruses from the permafrost, was based on scientific facts that were no less real and perhaps even more frightening. Having sorted out the real story that probably served as the first component in the creation of the ice lab myth, we return north to the cold lands of Alaska and Siberia. It is here that the second, more mystical and frightening part of the legend begins, the story of ancient viruses awakening from their slumber. And if the disappearance of viruses from the laboratory is the result of human error, then the threat from the permafrost is a consequence of global processes that we ourselves have set in motion. What is permafrost? It is a vast area in the northern hemisphere, about a quarter of all land, where the ground remains frozen for at least 2 consecutive years. In some places, such as Yakutia, this ice shell is up to 1.5 kilometers thick and hundreds of thousands, or even millions, of years old. Permafrost is not just frozen soil, it is a giant natural freezer, a cryochamber on a planetary scale. Everything that has fallen into this chamber during the last ice ages has been preserved in perfect condition. The remains of mammoths, woolly rhinoceroses, cave lions, ancient plants, and along with them, countless microorganisms, bacteria, archaea, and, of course, viruses. For thousands of years, this world was securely sealed. But today, in the era of global warming, this seal is beginning to give way. Permafrost is melting, and it is melting at an unprecedented rate. As it melts, huge amounts of organic material that has been frozen for hundreds of thousands of years is being released. As it decomposes, methane and carbon dioxide are released into the atmosphere, further accelerating the greenhouse effect. But that is not the only danger. Along with ancient organic matter, its microscopic inhabitants are also being released. Scientists have long wondered whether microbes that have been frozen for tens and hundreds of thousands of years can become active again. The first answers to this question were obtained in the early 2000s. Researchers from Russia, the US, and Europe began drilling into the permafrost and studying the extracted cores, long ice cylinders representing a slice of time. The results were astonishing. Viable bacteria were found in samples hundreds of thousands of years old. These microbes, which had been in suspended animation for an eternity, began to grow and multiply again when placed in favorable conditions in the laboratory. It was proven that bacteria can survive such prolonged freezing. But what about viruses? Viruses are not exactly living organisms. They are inert particles, a kind of biological nanorobot that only comes to life when it enters a living host cell. This characteristic, being inert outside the cell, theoretically gave them even more chance of surviving deep freezing. It was this hypothesis that formed the basis of the research that gave rise to the term zombieviruses and attracted worldwide attention. Science was on the verge of discovering a whole lost world of microbes, and the consequences of this discovery were completely unpredictable. The term zombieviruses sounds like the title of a horror movie, but it accurately describes the phenomenon that scientists encountered. These are not viruses that turn people into the walking dead. These are viruses that, despite being dead, or more accurately, dormant for thousands of years, are capable of returning to life and becoming infectious again. A group of French scientists led by Professor Jean-Michel Claverie of Aix-Marseille University became pioneers in this field. Unlike many of their colleagues who were searching for RNA or DNA from ancient pathogens in the permafrost, Claverie's team set themselves a more ambitious and risky task. They wanted to find and revive an entire functioning virus. To make their experiment safe, they decided to focus on finding so-called giant viruses. These viruses, such as mimiviruses, pandoraviruses, and petoviruses, are so large that they can be seen under a regular light microscope. But their most important feature is that they only infect single-celled organisms, specifically amoebas. They are completely harmless to humans and other mammals. Amoebas have thus become a kind of canary in a coal mine, an indicator that could show whether ancient viruses that infect eukaryotic cells are capable of remaining infectious. In 2014, Clavery's team announced its first success. They managed to revive a phytovirus extracted from a 30,000-year-old sample of Siberian permafrost. In the laboratory, this ancient virus, placed in a petri dish with live amoebas, immediately began to attack them and multiply, as if those 30,000 years of suspended animation had never happened. It was a scientific sensation. For the first time, it was proven that a complex DNA-containing virus capable of infecting eukaryotic cells could remain viable for such a long time. This experiment opened Pandora's box. On the one hand, it gave scientists a unique tool for studying the evolution of viruses. On the other hand, it became a vivid demonstration of the potential danger lurking in the ice. If an amoeba virus can survive, why can't others? In the years that followed, Clavery's team and other research groups continued their investigations. They revived several more types of giant viruses found in a wide variety of places. In the frozen stomach contents of a Siberian wolf, in clumps of mammoth wool, and simply in ancient layers of permafrost. The oldest of the revived viruses, Pandoavirus, discovered at the bottom of a lake in Yakutia, was estimated to be 48,500 years old. This is a staggering figure. This virus was active at a time when Neanderthals still walked the earth and slept through the entire history of human civilization, only to awaken again in the 21st century in a laboratory. Experiments with zombieviruses were irrefutable proof. Permafrost is not a graveyard for microbes, but rather their bedroom. And now, due to climate change, their alarm clock is ringing. The question is no longer, can they survive? Now it sounds different. What other viruses are sleeping there, and what will happen when they wake up? Jean-Michel Clavery has become a central figure in the study of ancient viruses. His work is a blend of courage, curiosity, and stark warnings. He is not just a scientist, he is a messenger, trying to alert the world to a potential threat that many prefer to ignore. His team's approach was methodical and cautious. They never set out to find and revive a virus capable of infecting humans. We would never take the risk of isolating a virus that is ultimately capable of infecting modern mammals, Clavery said in an interview. Using amoebas as bait was a brilliant move that proved the virus's ability to survive without posing a biological threat. However, Clavery himself understands perfectly well that his safe laboratory experiments are only a model. In reality, when the permafrost melts, no one will sort viruses into dangerous and safe. They will all be released. We see traces of many other viruses, he warns. If amoeba viruses are still alive, there is no reason why other viruses would not still be alive and capable of infecting their own hosts. These hosts could be anything from mosquitoes and birds to rodents and humans. Clavery's team's research has also shown how diverse the world of ancient viruses is. They have revived representatives of completely different families: pitoviruses, pandoraviruses, and mimiviruses. These viruses differ from each other in both structure and replication method. This diversity suggests that the ability to survive in permafrost is not a unique property of a single group, but is likely inherent in many types of viruses, including those that can be pathogenic to humans. In addition, modern methods of metagenomics, the analysis of all genetic material present in a sample, confirm Clavery's fears. In samples from permafrost, scientists are finding genetic traces of well-known human and animal pathogens, fragments of DNA from pox viruses, which include the smallpox virus, herpes viruses, and asfar viruses, have been detected. Although these are only genetic ghosts and not living viruses, their presence proves that they were there. And if conditions allowed giant amoeba viruses to survive, then some of their more dangerous relatives could also have been preserved. The work of Clavery and his colleagues has led to plans for an Arctic monitoring network. The idea is to track early cases of unusual diseases in humans and animals in Arctic regions. This would allow new threats to be quickly identified, isolated, and prevented from spreading beyond the region. In essence, it is an attempt to create an early warning system for pandemics caused by ancient pathogens. Jean-Michel Clavery's work is therefore significant in two ways. On the one hand, it expands our fundamental knowledge of the world of viruses. On the other, it serves as a loud and persistent warning. A warning that the past is not dead, but merely lying dormant beneath the ice. And our modern world, with its globalization and high population density, may find itself completely unprepared to face enemies we thought long extinct. Although the most high-profile experiments in virus revival have been conducted with Siberian samples, Alaska is also the focus of scientists studying threats from the permafrost. Like Siberia, this region is a vast repository of ancient life, and it is melting at an alarming rate. What real discoveries have been made in Alaska, and what do they tell us? One of the most significant events in the history of ancient pathogen research in Alaska was the exhumation of the remains of a woman who died in 1918 in the settlement of Brevig Mission. She was a victim of the most terrible pandemic in human history, the Spanish flu. Her body was buried in a mass grave in the permafrost. In 1997, a group of scientists led by Johan Kalthen obtained permission to exhume the remains. In samples of lung tissue, perfectly preserved thanks to the cold, they managed to detect genetic material from the 1918 H1N1 influenza virus. This discovery allowed scientists to completely sequence the genome of the Spanish flu, understand the reasons for its extreme virulence, and develop new strategies to combat future influenza pandemics. Although the virus itself was not revived, this case clearly showed that permafrost can preserve the genetic material of pathogens for decades. Other studies have focused on finding traces of even more ancient diseases. Fragments of smallpox virus DNA were found in a 300-year-old Siberian mummy, also found in permafrost. This suggests that the virus, which humanity defeated with great difficulty in the 20th century, could potentially survive in frozen graves. The outbreak of anthrax in Yamal in 2016 was another ominous warning. At that time, abnormal heat thawed the carcass of a reindeer that had died of anthrax decades ago. Spores of the Bacillus anthracis bacterium were released, infecting dozens of people and killing thousands of reindeer. This case showed how quickly and suddenly an ancient threat can become a modern reality. In Alaska, scientists from the University of Alaska and the Centers for Disease Control and Prevention, CDC, are actively studying the local fauna for unusual viruses. They catch small mammals, voles, squirrels, shrews, and analyze their tissue and blood. It is this work that has led to a discovery directly related to our investigation and adds another layer of reality to it. During this monitoring, a completely new virus, previously unknown to science, was discovered. It was named after the place where it was found, and its name echoes the original legend with which we began. It was named Alaskapox, the Alaska smallpox virus. This discovery proves that the Arctic regions are not just a repository of old threats, but also a cradle for new ones. The discovery of Alaskapox is a perfect example of how science works at the cutting edge. It was not the result of a search for a specific threat, but rather the result of systematic and methodical study of the wild. The story began in 2015, when a woman from the Fairbanks area of Alaska went to her doctor with an unusual skin rash that she initially thought was a spider bite. She also had fatigue and fever. Doctors were unable to immediately determine the cause, but samples sent to the CDC laboratory revealed something unexpected. It was an orthopox virus, belonging to the same family as the viruses that cause smallpox, cowpox, and monkeypox, but genetically distinct from all known members of the group. It was a new virus. Subsequent research showed that Alaskan pox appears to be a zoonotic virus circulating among small mammals in Alaska. Its DNA and antibodies to it were found in red voles, northern flying squirrels, and shrews. Scientists suggested that humans could become infected from these animals, possibly through domestic pets. For example, a cat that caught an infected vole could then scratch its owner, transmitting the virus. Over the course of several years, only a few cases of Alaskan pox in humans were reported. All were mild, and the patients recovered on their own. The virus did not appear to pose a serious threat and was not transmitted from person to person. It was more of a scientific curiosity than a public health concern. However, in early 2024, the situation changed. An elderly man with a weakened immune system from the Kenai Peninsula became the first reported victim of the Alaskapox virus. He died from a severe form of the disease. This case prompted Alaska health authorities to reevaluate their approach to the virus and issue recommendations for doctors and the public. It became clear that the virus could be deadly for people with immunodeficiency. The Alaskapox phenomenon is important to our investigation for several reasons. First, it proves that previously unknown pathogens capable of infecting humans are indeed circulating in the wild in Alaska. This is no longer a theory, but an established fact. Second, it shows how a virus that was considered relatively harmless can be deadly to vulnerable populations. Third, its very name, Alaskapox, creates a powerful informational link to the region, making any rumors about viruses from Alaska more credible. Imagine how this information is refracted in the mind of the average person, frightened by news of pandemics. A new smallpox virus has been found in Alaska, and it has already killed someone. This statement, while completely true, falls on fertile ground prepared by fears of zombieviruses from the permafrost. Now, all the pieces of the puzzle are in place. We have a real story about negligence in a laboratory and the disappearance of viruses in Australia. We have real scientific research on the revival of ancient viruses from the Siberian permafrost, and we have a real, new, and potentially deadly virus discovered in Alaska. It's time to put these pieces together and understand how a myth is born. Now that we have separated fact from fiction, we can reconstruct the process by which the myth of the 4 capsules from Alaska was born. This process is similar to alchemy, where disparate but real elements are combined to create something new, a coherent, frightening, and convincing story. At the heart of any enduring myth are real emotions and fears. In our case, these are fears of pandemics, exacerbated by recent global experiences, fears of invisible threats such as viruses, and a growing distrust of official institutions and science. These fears create fertile ground. Three main pieces of information, which we have analyzed in detail, fall onto this soil. The first piece is the Queensland incident. This is the story of the real-life disappearance of hundreds of test tubes containing dangerous viruses due to negligence. This story provides a factual basis for the myth. Yes, viruses can indeed disappear from a laboratory. It also provides a motive for concealing information, as the Australian authorities remained silent for 2 years. The second seed is research into zombieviruses. The work of Jean-Michel Claverie and his colleagues gives the myth a scientific veneer and an exotic, chilling element. Ancient pathogens, dormant in the ice since the time of the mammoths, are a powerful image. They appeal to our fear of the unknown past and of the forces of nature that we awaken. This element transports the setting from mundane Australia to the mysterious and mystical Arctic. The third seed is the discovery of the Alaskapox virus. This is a local connection that grounds the myth. It makes the story more concrete and plausible. It is no longer just some abstract Arctic, but Alaska, where a new and dangerous virus has been found. Now begins the process of alchemical synthesis, which usually takes place in the anonymous environment of online forums, social networks, and blogs. Details from different stories begin to mix and mutate. Simplification and dramatization. Instead of the complicated and boring 323 test tubes lost due to a bureaucratic error, we get the clear and ominous number 4. 4 capsules sounds like something special, something selected. The loss due to negligence turns into a mysterious theft with broken cameras and opened storage facilities. Merging. The Queensland laboratory moves to Alaska. The missing Hendra and hantavirus viruses are replaced by the much more intriguing ancient pathogens from the permafrost. Addition of details. Fictional details are added to make the story more convincing. Secret Level 4 facility. Scientists studied mammoth tissue. These details appeal to our stereotypes from spy movies and science fiction. The result of this process is a finished product. Where did 4 capsules containing ancient viruses disappear from an Alaskan laboratory? This story is perfect. It is easy to understand, evokes strong emotions, fear, curiosity, has a scientific basis, and hints at a conspiracy theory. It spreads easily because everyone who retells it feels like they are the bearer of secret, important knowledge. Thus, the Alaska myth is not just a lie. It is a symptom of our time, where information spreads instantly. Facts are mixed with fiction, and collective fears can crystallize into very convincing but completely unrealistic stories. Understanding this mechanism is as important as debunking the myth itself. The story of the missing viruses, whether fictional in Alaska or real in Australia, inevitably raises the question, how safe are we? How reliable are the systems designed to contain the most dangerous microorganisms on the planet? Laboratories working with dangerous pathogens are classified according to biosafety levels, from BSL-1 to BSL-4. BSL-1 is a typical teaching laboratory. BSL-4 is the highest level of isolation, designed for working with deadly viruses for which there are no vaccines or treatments, such as Ebola or Marburg. Scientists in these laboratories work in self-contained airtight suits. and the laboratories themselves are sealed boxes within boxes that are kept under negative pressure so that no particles can escape. The Queensland lab, like most government virology centers, operated at BSL-2 and BSL-3 levels. This is sufficient for working with viruses such as Hendra or hantavirus. Protocols at these levels are extremely strict. Restricted access, special ventilation, strict rules for handling and disposing of materials. However, the Queensland incident showed that even the most sophisticated technical system is vulnerable to human error. Protocols only work when they are strictly followed. Fatigue, rushing, understaffing, incorrectly completed paperwork. Any of these little things can become the weakest link in the security chain. This incident forced a rethink of approaches to biosafety around the world. It became clear that, in addition to physical protection, locks, sealed boxes, bureaucratic protection, a system of record-keeping and double checks, is equally important. Modern systems increasingly rely on automation, barcoding each test tube, tamper-proof electronic logs, and video recording of all handling of particularly dangerous materials. The goal is to minimize the influence of the human factor and make it impossible for hundreds of samples to be lost for 2 years. But there is another side to the problem: the growing number of high-level laboratories around the world. After the COVID-19 pandemic, many countries began investing in the creation of their own BSL-3 and BSL-4 laboratories to be prepared for future threats. On the one hand, this is good because it increases global preparedness. On the other hand, it increases the number of places where an incident could occur. More laboratories means more risks of accidental leaks or deliberate sabotage. Biosafety experts are talking about the need for global oversight and standardization of protocols. International inspections, common standards for staff training, and transparency in the investigation of any incidents are needed. The safety of one laboratory is the safety of the whole world, as viruses do not recognize national borders. Thus, while we peer into the mysterious depths of the permafrost, a real and everyday threat may be much closer, in a laboratory freezer down the street. And combating this threat requires not only high technology, but also the highest level of responsibility and discipline. We have examined the threat of human error and discussed biosafety systems. But what if the most significant and largest intruder into nature's cryopreservation facilities is not a human being? What if it is a process that we ourselves have set in motion but can no longer fully control? We are talking about climate change. Global warming is having a much greater impact on the Arctic than on the rest of the world. This region is warming 2 to 3 times faster than the global average. The consequences are visible to the naked eye. Glaciers are melting, sea ice is shrinking, and permafrost, which has been permanent for millennia, is beginning to thaw. Permafrost thawing is not just a slow warming of the soil. It is a dramatic process that changes the landscape. It leads to the formation of thermokarst lakes, ground collapses, and landslides. Entire sections of the Arctic Ocean coastline are collapsing into the water, releasing tons of ancient organic material. Unlike a laboratory accident involving hundreds of test tubes, megatons of organic matter containing billions of microorganisms are being released into the environment. This is the largest laboratory accident in the history of the planet, and it is happening right now. Professor Clavery and other scientists warn that this process is irreversible. Even if we stop all greenhouse gas emissions today, there is already enough heat stored in the system to keep the melting going for a long time. This creates a completely new, previously unknown risk factor. As the Arctic warms, it becomes more accessible to humans. Shipping is developing along the northern sea route, and the extraction of natural resources such as oil, gas, and minerals is beginning. More and more people will live and work in these regions. This means that the likelihood of human contact with ancient, reawakened pathogens will only increase. Imagine this scenario. During drilling or construction in an area of thawed permafrost, workers come into contact with an unknown virus. They develop an unusual disease. Since local medicine is not prepared for this, it takes time before the correct diagnosis is made. During this time, infected workers may return home to large cities in other parts of the world, spreading the virus through the global transport network. This scenario is frighteningly realistic. It does not require malicious actors or conspiracy theories. It only requires a combination of two factors that already exist: the melting of permafrost and increased human activity in the Arctic. Climate change, therefore, acts as a universal key that unlocks not one, but millions of capsules containing ancient viruses. And unlike a laboratory, there are no safety protocols or containment systems. Nature itself is conducting an experiment to release ancient life, and humanity risks becoming its main test group. The final question of our investigation is no longer whether the threat is real, but how we will respond to it. We began our journey with a simple and disturbing mystery. Where did 4 capsules containing ancient viruses disappear from a laboratory in Alaska? After navigating a maze of rumors, real incidents, and scientific discoveries, we have reached the end, where the original mystery loses its meaning, giving way to much deeper and more serious questions. The mystery of the ice lab turned out to be a myth. There was no secret facility, no stolen capsules. But like any powerful myth, this story proved to be truer than many facts. It became a mirror reflecting our most pressing fears and most real threats. It forced us to talk about things we prefer not to think about. We learned that the threat of laboratory incidents is very real. The Queensland case is not an anomaly, but a stark reminder that the human factor can undermine even the most sophisticated safety systems. Responsibility, discipline, and transparency are the only reliable barriers capable of containing the pathogens we keep in our laboratories. We have learned that the threat from permafrost is not science fiction. Scientists are indeed reviving viruses that are tens of thousands of years old. And although they are currently working with models that are safe for humans, no one can guarantee that pathogens capable of causing a new pandemic are not lurking in the melting ice. We have realized that the main culprit behind future biological threats is not a mysterious spy, but all of us. The climate change process we have set in motion has turned the Arctic into a planetary time bomb. And with each passing year, with every centimeter of permafrost that melts, its clock is ticking louder and louder. So what, in the end, is the secret? It has no answer because it was made up. But it leaves us with a real problem that has no solution yet. The story of the 4 capsules is a convenient and understandable image of the enemy. It is easy to fear a specific villain who has stolen a specific virus. It is much more difficult to fear systemic negligence, invisible microbes, and the long-term consequences of our own actions. The real mystery is not where the 4 fictional capsules disappeared to. The real mystery is what billions of people on the planet will do when thousands of real, unstudied, and potentially dangerous microorganisms begin to emerge from the permafrost. The question is no longer, where did the viruses go? The question is, what will we do when they appear? And no one has an answer to that question yet. Our investigation is complete, but the real story seems to be just beginning.